Okay, so this video is designed to help you with your test on the nervous system. This is not all that you need to know about the nervous system. This is just an explanation of some of the most important things, so make sure that you study your notes also. Okay, the nervous system has three major components: sensory input, integration, and motor output. Okay, so the first one, sensory input, that's going to be the senses. So you have sensory input where you see, when you hear, when you taste, when you feel a touch, anything where a nerve senses an impulse. And then the second thing is it's gonna take it to the brain. So you're gonna interpret that, or how you perceive it. So that would be how you think about it. And then the third thing would be when the nervous system has the ability to send information from the brain to an effector cell. Okay, so let me say that a little differently. The first thing is your brain can sense things. So the nerves can take an impulse to the brain. The second thing is your brain can perceive information. It can take information and identify what it is or think about what it is. Okay, and then the third thing is that your brain can send information, such as to glands or muscles, to tell them to react. So sensory input would be, for example, you hear something, and that's by the bending of the ears, the ear here. Okay, and then you perceive it. Oh, that's a dog bark. I'm sure that's a dog barking. Okay, so the motor output would be that you run away because there's a dog chasing you. Okay, the second thing you need to know is the parts of the neuron. So you want to be really familiar with the structure of the neuron as well as the function of the neuron. Okay, so these little guys out here are dendrites, and dendrites receive the information. So the dendrites are going to receive the information either from a stimulus, like it's on the end of your fingers, or receive the information from the neuron before it. So that would be the reception of signal transduction. Okay, the cell body, which is next, that's where the information is changed. That's where the information is put all together. Do you remember? If you remember the word summation, that's where the impulse is summated. Okay, and then the axon is where the information is sent down. So dendrites receive, axons send. The axon is wrapped in Schwann cells, and those are just protective insulating cells. And together, all the Schwann cells make up the myelin sheath. I think it's right there, the myelin sheath. Together, they make up a protective layer, and I told you in class to think about a rubber coating around a wire on your earbuds that you listen to music with. So Schwann cells make up the myelin sheath, and in between Schwann cells, there's these little gaps, and they're called nodes of Ranvier. And the impulse, instead of going straight down the axon like we would assume, jumps the nodes of Ranvier. And that the word for the jumping is called saltatory conduction. So as the impulse is propagated down the axon, it has a saltatory conduction motion, which would be jumping. Okay, the end of the axon has an axon terminal right here, and that's what's going to release the neurotransmitter to the next nerve. So the terminal releases the neurotransmitter to the next nerve. Okay, so this one, oh, it still says data question at the top, sorry. This picture is a really important picture. This is how a nerve sends a signal. So this is how the nerve actually sends the signal. The first thing it shows you is depolarization, and that's when sodium rushes into the axon. Okay, once it reaches this positive 30, the impulse, which we said is like a charge, travels down the axon. Once it's traveled down, the neuron is going to repolarize by sending potassium out. So 3 was potassium out. 4 is hyperpolarization. It's like a resting, it's where I don't want to say resting potential, but that's where the nerve takes a break. So it sends out so much potassium that it gets a second to take a break before the next impulse is sent. So this is signal transduction. These are the steps to signal transduction. Not very good writing there. Okay, so steps to signal transduction are depolarization. Sodium rushes out. Oh, sorry, sodium rushes into the cell. Depolarization, sodium rushes in. Action potential, that's when the nerve impulse travels. Make sure if you're writing about this, you say the nerve impulse travels, not the neurotransmitter. Repolarization is when potassium goes out of the cell. Hyperpolarization is when there's too much potassium out. Okay, and then back to resting, which is right here, at -70. And remember we said resting potential is established or maintained because of the sodium-potassium pump. So sodium-potassium pump maintains that -70. Remember to think about this as salty banana, and I told you that that's where the salt, or the sodium, it's not really salt, it's really sodium is on the outside, and potassium is on the inside, like a salty banana. Okay, one more thing from this picture that isn't labeled on the picture, but threshold is the -55. So threshold, if I had another line, I would have it there. Threshold is the point of no return. That's the all or nothing. It's got to reach threshold and then it's going. So one thing about the nerve, once it reaches this threshold point, the nerve impulse is going to travel. It is going to go, and it doesn't matter how much of the nerve is depolarized. The nerve impulse isn't going to travel any faster or only halfway. The nerve impulse is going to travel if it reaches -55, and there's no it's going to only get to +30. That was when the nerve impulse travels. So when something has a small feeling, it would only have one action potential like that. But if something hurts, it would have a lot of action potentials. So let's see if I can draw this. So this would be something in pain. And this would just be just a slight touch. So you want to know -70, -55, and +30. So +30 is your action potential. Okay, and here's the same thing. It's just showing you that the sodium potassium go in through gated ions. Oh, going back. Sodium potassium right here. You can see the sodium going in. So this is dipole. No. Let's start over. This is 1 right here. Sorry. So you can see this is resting potential. So resting state, the sodium and the potassium channels are closed. So you can see all the gates are closed. What's maintaining this, if I could add to this picture, it would be the sodium-potassium pump is maintaining that. Okay, so depolarization, that's when sodium floods in. And you can see the potassium gate is still closed. But this is just showing continuous depolarization. You've got more sodium coming in. Okay, so the next thing on 4, it says fall. I would call this repolarization. So we're right here on the graph, and that's where potassium leaves the cell. So if you can remember salty banana, it's opposite of that. So the sodium goes in and the potassium goes out. Okay, and then this would be hyperpolarization, where too much potassium goes out. So this is 5 right here, hyperpolarization. So you definitely want to know the steps to that. Okay, so what happens at the end? Once the nerve impulse gets to the end of the neuron, so here's the nerve impulse at the end of the neuron, and remember, nerve impulse is like the charge. Once the nerve impulse has traveled to the end, calcium is going to flood in, and it's going to surround the vesicles of neurotransmitters. So the neurotransmitter was already at the terminal of the axon. Okay, so calcium comes in, surrounds the vesicles, and then the vesicle fuses with the axon cell membrane. So you see how the vesicle fused right there? How the vesicle is going to release neurotransmitter out into the synapse. So the nerve impulse got to the end, calcium came in, calcium fused with the vesicle of neurotransmitter, the neurotransmitter was released to the outside, Okay, now look right here. The neurotransmitter, look in this little bitty square. The neurotransmitter binds to the receptor on the postsynaptic neuron. This is the presynaptic. This is the postsynaptic. So the neurotransmitter binds to the receptor on the postsynaptic terminal, and when it binds, it opens up the receptor, and that's going to allow sodium to flood in the postsynaptic neuron. So all it did was, when acetylcholine binds, right here, when acetylcholine binds, the next neuron is depolarized. Okay, so this is a pretty tough concept. Think about, let's go through it real quickly. The first thing is that the nerve impulse gets to the end. Calcium comes in and surrounds the vesicles. The vesicle is fused to the terminal, which releases neurotransmitter into the synapse. The neurotransmitter binds to the receptor protein on the postsynaptic neuron, which opens up the channel. The channel allows sodium to flood in, and the sodium depolarizes the next neuron. Okay, so take a second, pause the video, and see if you can write down those steps, because that's hard. Okay, so how does signal transduction work with neurons? So you know the steps to signal transduction, and they are reception. You should know this easy. Reception is when the ligand binds and the protein receptor or the G protein changes shape. Transduction is where the message is converted on the inside of the cell. And then the response is where a cellular response occurs, such as protein synthesis or something like that. So how does this have anything to do with neurons? Well, neurons talking to neurons. That's all signal transduction is. And this is signal transduction. Okay, so here's how it happens. Acetylcholine binds to the neuron. So this would be like the postsynaptic neuron, because this would be the synapse. So you can imagine here is the terminal of the axon before. So acetylcholine was released, and it binds right here. That's reception. Okay, well, when it does, the channel opens up. So reception, I said, was when the ligand binds and the protein changes shape. So acetylcholine binds, the channel opens. Transduction is that sodium is allowed to flood in, and that depolarizes. So we've taken the original message, which was this one, we've taken the message and we've changed it to something the cell can understand. That's what transduction is. And the response is simply that the neuron moves the impulse down. So the nerve impulse travels down. So step one, acetylcholine binds. Step two is that sodium floods in and the nerve is depolarized. And step three is that the nerve impulse travels down the axon. Okay, so now we're talking about the different sensations we have. And you know you have touch and taste and smell and hear, and you've known that. So what does this depolarization mean to your brain? So like when I touch something, I feel that I've touched something. But when I hear something, I know that I've heard something. But all we're talking about is these nerves being depolarized. So how does my brain interpret whether it's a sound or a touch? And the difference is that it goes to different parts of your brain. So if you look at this picture, and I just found this on Google, but it's showing you that the temporal lobe, so a certain location, is going to do hearing. So the temporal lobe does the hearing, which is this one. But the occipital lobe, which is this one, does vision. So the neurons take the nerve impulse to different parts of the brain. And you don't have to know what parts of the brain do what, but just understand that the impulse is simply that depolarization or that charge traveling down, and then it has to go to the different parts of the brain for us to interpret. So this is how we interpret. Okay, and this is sensation. So I thought this picture was really neat because it shows here are the dendrites, and they are going to receive the sensation of touch. Okay, and then I think this one shows that this is actually pain right here. And so it's going to receive the sensation, and then it's going to travel from that dendrite down, and then it's going to go to the brain so that you can interpret what it is that has been depolarized. So when you touch something, these dendrites right here, just all they do is receive the message so that the nerve can be depolarized. And I just thought this picture was neat because it shows right here, this was a light touch, and this one's pain, and this one is a strong touch, and this one is temperature. And so you can see that there's nerves on the under the surface of the skin, and there's nerves right here on this hair. So when a hair is pulled out, it hurts. It sends a sensation to the brain. Okay, so the first sensation we're going to talk about is the sense of hearing. So sound waves come in. Here they are coming in. You hear something. Okay, and they're going to vibrate this tympanic membrane right here, and that's what we commonly call the eardrum. And that's going to vibrate these three bones, malleus, incus, and stapes, and you don't need to know the name of those, but the vibrations vibrate the eardrum, which vibrate the bones, which are going to vibrate the cochlea right here, which is important for you. Okay, so the cochlea has inside it a fluid called perilymph, and it also has hairs inside it. So when the cochlea vibrates, the hairs vibrate the perilymph, which will bend. So when and this is your cochlea. Oh, I misspelled that. C-O-C-H-L-E-A. Okay, so remember that has inside it perilymph and hairs. So when the vibrations come in, it's going to bend those hairs. And when it does, it sends an impulse to the brain. Here's your brain. So I'm going to do it again in yellow. So the sound waves come in and vibrate and bend hairs. So there's where the hairs are bending in the cochlea. And when the hairs bend, it depolarizes this auditory nerve. And the auditory nerve takes the message to the temporal lobe of the brain right there. So that didn't work out like I was thinking it should have. There we go. They take the message to the auditory part. There, your brain can interpret what the sound was. So the key thing here is that hairs are bending, and that's causing depolarization. So imagine there's a nerve right there, which there should be in the picture. When the hairs bend, it causes depolarization in the membrane of the auditory nerve to create the action potential. And remember, the action potential is just electrical energy traveling down the nerve. And it's all because the perilymph bent that hair. Okay, taste is the same thing, except instead of having mechanical receptors right here, you have chemoreceptors. And those chemoreceptors taste the chemical of the food, and that's going to depolarize this, send the message to the brain. So taste is pretty simple. Chemicals cause the depolarization, so remember that, of the different neurons. Okay, and then smell, exact same thing. The smell comes in, chemoreceptors inside your nose. And so that's going to, right here, you see how it depolarizes it and sends an action potential to the brain. So that would be smell. Your notes tell you that 80% of taste is actually smell. That's just an interesting fact. Okay, so sight. One of the coolest things that we as humans can do. We're going to take in light waves this time instead of sound waves. And the light waves are going to hit the retina, which is that yellow part right there. And the retina has on it protein receptors, which are rods and cones. So these protein receptors, they're photoreceptors. They receive light. Rods are going to be for black and white. While cones are for color. So you can see, and I told you to remember that cones are for color because the the orange cones in the middle of the road. They're orange. So cones are for color, rods are for black and white, and these are receptors on the back of the eye. Your notes tell you that the rods have a pigment in them called rhodopsin, and that's a pigment that allows it to see the black. It's a combination of retinal and opsin proteins. A change in the shape of the rod. So when the light comes in, I like to draw on this thing, can you tell? When the light comes in, it's going to change the shape of the rod, and that depolarizes the neuron. Okay, so the next part of the notes is right here, and it tells you about locomotion, and it tells you that there's two things working against the locomotion, and that's friction and gravity. Okay, so this part of the notes is pretty easy. The animals in the water, because of the buoyancy, there's not much gravity that they're dealing with, but they're dealing with lots of friction as they travel through the water. So in order to overcome this, these animals have adapted, and they have a fusiform, or a torpedo-shaped body. Okay, the second one would be animals on land, like us, and we don't have much friction up against us. However, we do have a lot of gravity up against us. So we have evolved or adapted to overcome this by muscular limbs. Those are our limbs, muscular limbs. Okay, and then animals that fly, they have both oppositions. They have the opposition of friction and gravity. So in order to overcome this, they have hollow bones, so they're really light to overcome gravity, and they have these feathers that have allowed them to fly. Okay, so how we flex muscles, and I think this might be the hardest part. There's just so many details to this part of the notes. Okay, so the nerve impulse gets to the end. You already know this part, but we'll review it again. The nerve impulse gets to the end, and calcium floods in and pushes the vesicle to the terminal, and then the neurotransmitter is released into the synapse. And then remember how the synapse binds to the receptor? You got all that. Well, this time the neurotransmitter is specifically acetylcholine. It shows you right there. Acetylcholine binds to the receptor. When it does, the receptor opens so that sodium can flood in. You know that story. Okay, so the nerve impulse travels to the T-tubule and down the T-tubule. And when it does that, the sarcoplasmic reticulum, which is, the way I look at it, is smooth ER but in the muscle cells. The sarcoplasmic reticulum releases the calcium. Okay, so calcium goes into the cytoplasm. Calcium binds right here to the tryptophan complex. So let's take a second and look at what this is. Okay, the yellow or orange, that's actin. Okay, and then there is another protein filament. I'm going to do mine in green, called trypomyosin, and the trypomyosin is a protein filament wrapped around it, and then there's the tryptophan, is another little protein on it, which you see there. So there is actin, which is the yellow, trypomyosin, which is the thread, and then the tryptophan, which is the little protein. Okay, so calcium is going to bind, and you can actually see it binding right there. Calcium is green. Calcium is going to bind to the tryptophan complex, and when it does, it moves that thread, the trypomyosin thread. It moves it over. So when calcium binds, trypomyosin moves. Well, when the trypomyosin moves, it frees up the binding site for the myosin. So there's two kinds of filaments. There's actin and there's myosin. So the myosin is in purple. Okay, so when trypomyosin moves, it frees up the binding site so that myosin can now bind to actin, and that causes the muscles to flex. So this actin and myosin slide, and that causes the muscle to flex or shorten or condense. About that time, acetylcholinesterase will degrade acetylcholine up here. The channels will close and the whole process will end, and that's to flex one muscle. And I believe I have it here shown a little bit bigger. So remember I told you this part, the yellow beads or orange looking beads, that's the actin. Okay, and then the thread, oh, it's labeled up here. The thread is the tryptomyosin, and then the purple thing is the tryptonium. So the calcium binds right here to the tryptonium complex. It moves the tryptomyosin, so now the myosin can actually bind in the actin. So myosin binds to actin, and that's what causes the two protein filaments to slide together. Okay, so pause the video, and these are just some things that you need to know. Some things I didn't mention, some things I